connect the phospholipids on both sides of the membrane. They connect them in the hydrophobic interior. And so again, we had a phospholipid over here. Here's my phosphate. Here's my fatty acid tails. If I've got another one on the other side of the membrane, again, because you've got this regular zigzagging, this regular bending and kinking, they can actually form bonds between the fatty acid tails, and that produces what's known as a lipid monolayer. And so here again, here's my phosphate head, here's my glycerol, and here's the fatty acid tails. They're joined together in the hydrophobic interior of the membrane. That's even more heat tolerant than the one above it. And so for the extreme thermophiles, for the organisms that are living at 400 degrees Celsius in the hydrothermal vents at the bottom of the ocean, they have these kinds of membranes because they're extremely heat tolerant. They don't melt at the high temperatures. Now these regular structures are given a name. If you have the bilayer like you have up here, this is called a phytonal, where we have this repeated isoprenes over and over and over again. And if they're bonded together, it's called a biphytonal. But again, the idea is by having this kind of structure, by having these repeated regular kinks, the phospholipids can pack really tight together, and that increases their heat stability. And so we see this a lot in the thermophiles and especially in the extreme hyperthermophiles. You'll even get these little monolayers like you see here. Now, as I mentioned, there's going to be the 4 different phyla of the Archaea. The Archaea is the most sort of diverse. The current Archaea contains sort of the extremophiles. The Nanoarchaea are small and parasitic, and we don't know anything else about the Chorioarchaea except that they exist. So very quickly, if we look through some of the groups of the Archaea, you have the halophiles. These are the salt-loving organisms. And they have a number of sort of adaptations that allow them to live in environments such as the Great Salt Lake, the Dead Sea. This is Lake Zoom, which is in Egypt. And these have extremely high concentrations of different types of salts. And in some cases, they also have very high pHs, but it's usually the high amount of salt that is the issue. Because what happens is when you have an organism that lives in a very high salt environment, it's always in danger of losing water to its environment. Because the environment around the cell is hypertonic to the cell. So you have essentially this condition here, where the cell is always in danger of losing water to its environment. Because there's so much salt outside the cell, there's not as much salt inside the cell. And so the water inside the cell will move out to try and dilute out the environment around it. And that's just kind of a quick trick. If you always want to remember what it means, you probably learned hypertonic, hypotonic, isotonic, and things when you were in general biology. But just remember, water always moves to dilute. So if you have more stuff outside the cell, you have more stuff outside the cell, more salt outside and less salt inside, the water inside the cell is going to move out to try and dilute out the environment. You have the opposite situation here in a hypotonic, where you have more salt inside the cell and less salt outside. So in this case, if there's less salt outside the cell, it's going to move into the cell to try and dilute out the interior of the cell. And so the cell will blow up like a little balloon and then burst eventually. But for the halophiles, they live in this type of environment. There's so much salt, so many solutes around them that they're always in danger of losing water to their environment. And so they have a number of mechanisms, like a very thick cell wall. They have cell membranes that are less permeable to water. And they also will sometimes pump in other solutes. So let's say, for example, there's a lot of sodium or sodium chloride outside the cell. The cell may try and balance some of that by increasing potassium or something inside the cell, something that's a comparable solute or comparable salt. But in general, the cell doesn't want too much salt in the cell because that will damage the DNA, it will damage proteins. So they don't want to have too much salt, but they also don't want to lose all their water to the environment. So the halophiles have, sort of a number of challenges. Having too much salt inside the cell is bad. Having too much salt outside the cell is bad. And so that to form methane. You have a variety of different